Thank you for joining us today. I'm Francesca and I work in the Obs and Gynae department at the Addenbrooks, and I will be chairing today's event, which is part of the Early Researcher Seminar Series hosted by the Cambridge Reproduction SRI. I would like to say a few things before we start. First of all, we would like to thank the volunteer coordinators for organizing this series, to all the speakers, and also Christina, the SRI coordinator who put all this together and makes the events run smoothly. As you all know, the Cambridge Reproduction SRI is an initiative that brings together researchers from across the university who have an interest in any aspect of reproduction. It's easy to join, so the membership is open to all staff and postgraduate students. And you can just download the membership form in the SRI website and email the form to Christina. Christina maybe can put the link in the chat for everybody. So this is the last seminar of the academic year, but they will start again in the autumn. So postgraduate students and postdocs who would like to participate, please just contact Christina and let her know. The seminars are usually held online on the second Thursday of each month at 1 p.m. And the details of the coming seminars will be on the SRI website. So we have three presentations today, so we better get started. Each of them will be 15 minutes and we'll have question and answers at the end. So now, please mute your microphone, and then at the end, if you have a question, just put it in the chat or unmute the microphone and just ask the speakers. So we will start with our first presenter, Jake Thomas. He is a PhD student under the supervision of Naomi McGovern in the Department of Pathology, and his presentation is entitled Phenotypic and Functional Diversity of Human Placental Macrophages. Over to you, Jake.
Okay, so yeah, thank you for the introduction. My name's Jake. I'm a final year PhD student in pathology in the McGovern lab, and I'm going to talk to you today about some of the work we've been doing looking at human placental macrophages in the first trimester of pregnancy. So in our lab, we're really interested in the placenta, an organ which I'm sure many of you are familiar with. In my project, we look in the first trimester of pregnancy, between 6 to 12 weeks estimated gestational age. The placenta is a unique organ in the sense that it forms the barrier between the fetus and the mother during pregnancy. It's entirely fetally derived, and it's composed of a villous tree-like structure with these finger-like projections which embed into the lining of the uterus during pregnancy, which is referred to as the decidua. And the placenta is a really interesting organ because of the fact that its unique position. It has to carry out many roles, and to some extent it can be seen as having the functions of many different organs that would be carried out in adult life. So facilitating gas exchange, excretory functions, metabolic and absorptive functions, as well as signaling and acting as a physical barrier to infectious agents, which could infect and cause congenital disease to a baby during pregnancy. If we take a cross-section of a villus, we can have a look at the number of different cell types that are present within the placenta. It is surrounded by this layer of specialized epithelial-like cells called trophoblasts, which are unique to the placenta. And within the stroma, there are a number of different populations, including fibroblasts, capillaries, and Hofbauer cells, or HBC. And Hofbauer cells are these tissue-resident macrophages. So what are macrophages? Macrophages are white blood cells or immune cells that are found in all different tissues throughout the human body. And typically they're associated with driving inflammatory responses, so responses to infection, the recognition and the killing of microbes when you become infected with them. However, over the past few decades, we've become more and more aware of the fact that these resident cells have more specific roles in tissue homeostasis, so ensuring that the tissue that they reside in functions properly and having essential roles in making sure that tissue does what it's supposed to do. And what we've realized is actually that each of these functions is highly specific to the tissue in which a macrophage resides. So for example, the microglia, which are macrophages in the brain, have important roles in generating neuron synapses, and alveolar macrophages in the lungs help clear surfactant. So very little is known about Hofbauer cells and their functions, and we want to understand what their functions are in homeostasis, so the roles they have that are specific to the placenta, and how they might help respond to infectious challenges in the placenta as well and protect the baby during pregnancy. So the main aims of my PhD thus far have been trying to understand what the roles of Hofbauer cells are in homeostasis and in response to infection. So to study Hofbauer cells, the first thing we have to do is be able to find them and identify them. We chiefly do this by flow cytometry. So this here is showing the expression of CD45, a leukocyte marker, and CD14, a myeloid cell marker. So the CD14 positive cells we very early on saw were heterogeneous for the expression of HLA-DR. Now this was quite surprising to us, as HLA-DR is seen as a canonical marker of macrophages. And we hypothesized that due to the unique localization of the placenta, some of these cells might be maternal in origin and contaminating our preps. So to figure this out, we use HLA allotype antibodies that can be used to identify maternal and fetal cells in donors that have an HLA mismatch between mother and fetus. And in this donor, the fetus is A3 negative. So we know the DR negative cells marked here with the red box are the true Hofbauer cells, and the DR positive marked with the blue box are a novel population of cells that we refer to as PAMM, or placenta-associated maternal monocyte/macrophages. And we can use whole mount immunofluorescence to look at these cells. So here we have one of these projections, one of these villi in the first trimester placenta. We can see the Hofbauer cells in the center of the villi marked in red, and then one of these PAMM in green stuck on the outside, which is exactly where you'd expect to see maternal cells, as that is the part of the villus which is in contact with maternal blood. And so this method for distinguishing these cells is reliant on this HLA mismatch between the mother and the fetus. So this doesn't happen all of the time. So the first thing we wanted to do was distinguish Hofbauer cells from PAMM in a more reliable fashion without relying on this mismatch. So to do so, we looked at publicly available single-cell RNA sequencing datasets, this one published in 2018 from Cambridge, and within which there was a population of Hofbauer cells and PAMM, which we could identify using the markers CD68, which marks all macrophages, and HLA-DR, which we know marks PAMM. So what we did was we generated differentially expressed genes between these two clusters to try and identify useful markers that might help pull out these populations by flow cytometry for functional analysis. And this is the plot of the differentially expressed genes. As a quick summary, these dots or genes that lie here away from this central line on this side are enriched in Hofbauer cells. These genes up here are enriched in PAMM. So we can see HLA-DR genes are enriched in PAMM, and then a number of candidate genes which are enriched in Hofbauer cells. So we took some of these genes and we performed flow cytometry to try and find an optimal way of gating these cells in our flow cytometry experiments. And I've showed 3 different markers here: CD64, CD206, and folate receptor 2, all in combination with HLA-DR. And folate receptor 2 gave the best separation. So this is the gating strategy that we've now used and developed to isolate a pure population of Hofbauer cells, which we use going forward to try and work out what they do. But that's not really the end of it. In doing that analysis to find true Hofbauer cells, it yielded some quite interesting results within our PAMM populations. What we can see marked here in green and in orange is that actually there was a considerable degree of heterogeneity within these maternal cells that we wanted to try and resolve before we went into working out what these cell types do. The first of which, these PAMM2 marked in gold, we did some analysis to work out they're actually decidual macrophage contaminants. So they're actually maternal in origin. They don't actually reside on the placenta. So I'm not going to talk about these guys anymore. The green cells, we did flow cytometry and we were able to find they were heterogeneous with CD9 as well as HLA-DR. So it looks like there's 2 populations within this PAMM1 population. And so in adult tissues, these cells are adult macrophages. We know that macrophages are derived from monocytes. So we predicted that one of these 2 populations might be actual blood monocytes or monocytes within the intervillar space of the placenta. So to test this, we added CCR2 to our panel, which is a marker for circulating blood monocytes. And we were able to identify 2 populations shown with the gates here in green and blue. Green, which we're referring to as PAMM1A, are these macrophages unique to the placenta. Blue, PAMM1B, are monocytes. And we were able to isolate these populations and look at them under the microscope to verify what they actually are. So Hofbauer cells, PAMM1A and PAMM2, all have this typical macrophage morphology, this sort of fried egg morphology, if you will, very vacuolar. PAMM1B looks strikingly different, much smaller, and they have this very, very typical lobe round nucleus, which is a classic feature of monocytes. And then as a general overview, we can look at the relative abundances of these populations. So Hofbauer cells form the vast majority of CD14 positive cells in our preps, but PAMM1A and PAMM1B do contribute significantly to the total number of CD14 cells that we look at, around 10% each. And this is super important because actually when people have studied Hofbauer cells in the past, they take this entire cell fraction as true Hofbauer cells. So they would essentially be looking at all of these cells, assuming they're one population, when in reality, they're actually four. So if you want to study these populations, we now have the tools with which to do so. We can pull them out properly. And so one thing we wanted to look at a little bit further before getting into the Hofbauer cells was looking at these PAMM. We know that we have two populations of PAMM that reside on the surface of the placenta, monocytes that give rise to macrophages. And we wanted to see what genes get turned on as they become macrophages, and therefore might give us some idea about what these macrophages are doing on the surface. So to do this, we use single-cell RNA sequencing trajectory analysis, going from monocytes in blue to macrophages in green. And we can walk along this trajectory computationally, and we can look at the genes that get switched on or off to get an idea about what the cells are doing when they're fully differentiated. And so this is an example of such a plot where going from left to right, we're going from PAMM1B monocytes to PAMM1A macrophages. And red is where the gene is highly expressed, and blue is where the gene is lowly expressed. So this first plot is a whole set of genes that are being downregulated during differentiation, including typical monocyte genes such as IL-1 beta and S100A8. Conversely, we can look at genes that are upregulated during this differentiation, including CD9, CD63, CD68, and TREM2, all of which are typical macrophage genes. And then we can use some of these marker genes, so in this instance, CD9-HLA-DR, to look for these mature, fully differentiated PAMM1 on the surface. We're able to really nicely show HLA-DR, CD9 double positive cells embedded in the lining of the trophoblast in the villi, exactly as we would expect from our transcriptomic data. And so we also wanted to look a little bit beyond just gene expression, but also transcription factors, the proteins that regulate patterns of gene expression throughout this differentiation progress. And what was really striking to us was the fact that they were seeming to upregulate a transcriptional program which is associated with lipid metabolism, particularly the transcription factor PPAR gamma. And this was really interesting to us because we had access to some transmission electron microscopy taken by Graham Burton many years ago now, showing macrophages on the surface of placental villi that were heavily loaded with lipid drops labeled here in yellow. So it was really nice seeing this overlap between the transcriptomic data and the morphological data we could see by microscopy. We validated this via flow cytometry using BODP. BODP is a dye that stains lipid bodies or lipid droplets within cells. So we could show that PAMM1A were highly enriched with lipid droplets compared to these other macrophage populations. And lipid droplets and the upregulation of these transcription factors are known to be associated with the uptake of cellular debris in macrophages. So what we've predicted is that these cells are sitting on the outside of the placenta and are helping to clear any damage that occurs to it during healthy pregnancy. And it's well established that during healthy pregnancy, you do get damage to this layer. And with this prediction, we expected the PAMM to also produce more other, I guess, proteins and markers that would help repair the tissue. One of which is MMP9. We were able to show that MMP9 is highly produced by PAMM1A in comparison to the monocytes, but also fibronectin. So this is a matrix metalloprotease which remodels tissue, and this is an extracellular matrix protein which helps rebuild tissue. And so what we think is happening here is that PAMM are migrating onto the tissue there and having roles in the repair and the regeneration of the trophoblast layers that form the outside of the placenta through the uptake of cellular debris upon damage, and then through the laying down of MMP9 and fibronectin to help remodel the tissue. So that's a sort of overview of what we think their roles are in homeostasis. So now what I've shown is that we can isolate both Hofbauer cells and PAMM populations from our preps. We have gating strategies to do so. And this is allowing us to do functional assays now and going forward to elucidate the roles of these cells, both in homeostasis and in response to inflammatory challenges. The first thing we wanted to do was put these cells into culture, into dishes, with no stimulation, and see what cytokines they make. This gives us some idea about what the cells are doing at a steady state. And it was really interesting to see that Hofbauer cells produce a number of these factors that are involved in tissue remodeling, so MMP9, TIMP1, IL-1RA, as well as the growth factors VEGF-alpha, osteopontin, and fibroblast growth factor 2. Osteopontin and VEGF-alpha are particularly interesting to us because they're known to have pro-angiogenic properties, so they help the development of new blood vessels, which is really important in the placenta because at this time it's growing very rapidly and it needs to be very highly vascularized to promote oxygen transport to the fetus. And so moving past their roles in homeostasis, we wanted to look at their potential roles in inflammation and in infectious challenges. Some of the ways that macrophages and other immune cells recognize microbes is through the binding of specific receptors on their surfaces. One such family of receptors are the toll-like receptors, or TLRs, which each recognize a distinct pathogen-specific product. So what we did was we stained Hofbauer cells PAM1A and PAM1B for a range of different TLRs via flow cytometry to see if there were any differences between these populations. Hofbauer cells were found to very highly express TLR6, whereas the two PAM populations did not. TLR6 is known to bind bacteria lipoproteins, and Hofbauer cells also express very high levels of TLR7, more so than PAM1A and PAM1B. So then to back up this data, we did a similar experiment to this one here, where we put the cells in culture, but instead of leaving them on their own, we stimulated them with a range of agonists specific for each of these receptors. And so what we look at here is whether or not I've cut off all of the names of these proteins, so I'm going to have to tell you what they are. But in red are cytokines that go up, and in blue cytokines that go down. And what you can see is that in stimulation with FSL1 in Hofbauer cells, which is a TLR6 ligand, you get the upregulation of this is IL-6 and this is GM-CSF. You're going to have to believe me, I'm afraid, which we do not see in PAM1A because they don't express TLR6. But interestingly, when stimulated with the TLR7 agonist in Micromod, there was very little change in Hofbauer cells, but very apparent change in PAM1A, particularly in making IL-1 beta. This was interesting considering Hofbauer cells do express TLR7. So what we think is happening here is that although neither of these cells would be sufficient on their own to protect against either bacteria or single-stranded RNA viruses, in combination they can provide an effective barrier to infections at the placental surface. And then finally, one of the things we wanted to look at was whether or not these cells were capable of killing bacteria. And so to kill bacteria, you need to be able to do two things: internalize them and then kill them once they're there. So the process of internalization in macrophages is called phagocytosis. And what we did was we incubated our macrophages with fluorescent beads, and we can report the proportion of cells that uptake these fluorescent beads. And Hofbauer cells are really, really phagocytic. They eat a lot. PAM1A eat far less. We also then wanted to look at the internal pH of these cells. The pH can have an effect on how well bacteria survive once they're inside cells. And we used a pH-dependent probe bound to beads that then get internalized by the Hofbauer cells or PAM, and then we can look at the color of the beads to work out the pH within the cell. And what I hope you can see here from the representative images is that the Hofbauer cell has this kind of orangey color, whereas the beads in the PAM have this more sort of greenish, yellowish color, indicating that the pH within Hofbauer cells is lower than the pH within PAM. And then finally, we actually did some co-cultures and bacterial killing assays, and we were able to show that for two species of bacteria, Lactobacillus and E. coli, Hofbauer cells and PAM1 can kill these bacteria to similar extents. This is despite the fact that they have differences in pH and their phagocytic ability. So at the moment, we're not quite entirely sure the mechanisms that underlie the killing in these two different bacterial types. But the take-home message is that they do display microbicidal activity in vitro. And so with that, I will conclude and wrap up. Our first trimester Placental digests contain both fetal and maternal myeloid cells. This is a really important take-home message for anyone who wants to study Hofbauer cells or PAM in the future. You have to do these flow cytometry gating strategies to pull out the pure populations. Leading on from that, Hofbauer cells can be identified as folate receptor 2 HLA-DR negative macrophages, which we predict to have roles in tissue remodeling and angiogenesis in a steady state, so helping the placenta to develop and grow. The maternal cells have three populations: our macrophages, monocytes, and our decidual contaminants. And we think that PAMM1A in the steady state are having roles in the repair and regeneration of the trophoblast layers during healthy pregnancy. And we've shown that both Hofbauer cells and PAMM1A are capable of recognizing and killing microbes, but they show distinct responses to stimulations, which suggests that they have complementary roles in defense of the placenta, and they work together to protect against a range of pathogens, which could otherwise cause devastating consequences for the fetus. And in terms of future directions, where we want to think this research to go in the future, we're interested in understanding how the properties of these cells change throughout gestation. So all of this work at the moment is undertaken in the first trimester of human pregnancy. We want to see how these populations change by the time you reach term, so 40 weeks as opposed to 6 to 12 weeks. And then what are the roles of Hofbauer cells and PAMM in the prevention or permission of different trans-placental infections? So some infections such as Listeria or Zika or cytomegalovirus can cross the placenta, whereas others can't. And presumably that permissibility is dependent on the functions and the roles of both Hofbauer cells and PAMM. So we want to try and unpick the variability there in these different infections. And then finally, if anyone is interested in the work that I've shown today, we published it earlier this year, so you can go away and you can look up this paper if it's something you're interested in. And so with that, I'd like to thank a few people. Naomi in my lab has been really, really helpful in this, giving me really amazing supervision. Within the Department of Pathology, Ashley Moffat and Andrew Sharkey also helped a lot with this project, as well as Ritu Rainey, who liaised with people at the hospital to get tissue. And then at the hospital, importantly, Diana Myrna for consenting patients and for all the sample donors who donated samples to make this work possible. Thank you.